So it's very exciting for me to get the chance to talk to you all this evening. The main reason that we do the work that we do, at least from my perspective, and I know many of my colleagues, is we're trying to do things that will explain problems and help families, and we can produce these outputs in a variety of different ways. But one of the hardest things to do is to translate that information back very easily, and it's not easy for people such as yourselves, the families in general, doctors, teachers, to be sitting reading our scientific papers. We have to write them at a certain level so that they get past our reviewers. And so I always look forward to talks like this to try to explain some of what it is that we're doing and translate it into a way that hopefully, we'll see how I go, that you can understand. And if you have a child, or you know, or you're teaching a child, or you're trying to deal with some of these kinds of issues, maybe you'll walk away with a potentially better understanding that you can use as ways to actually guide your thinking about how to help children such as this. And our goal is to develop explanations of some of these phenomena so that we can develop interventions. As I always like to say, you can't fix something until you know how it works. So that's really the goal here. And so what I'm going to try to do is talk to you about a study that we've been doing for quite a while now. It's actually several studies that are combined. And it's been very striking that the thoughts and the predictions that we made in the beginning seem to be coming out sort of as we expected they would. And it may be that it's an overstatement to say that there's one explanation for several different disorders, but it does appear there may well be a sort of a final common pathway to some of these developmental phenomena, and that may really help us explain how they're happening and what kinds of interventions might make sense, and as we develop them, hopefully, that they'll be widely applicable. So let me start by talking to you about the disorders, and if you've been here to the Mind Institute before, you will have heard of at least some of these. I'm going to talk about, one of them I'll talk about is Fragile X, and I'm going to talk about girls who have the full mutation. So what does that mean? Fragile X is caused by a mutation on the FMR1 gene, which is located on the long arm of the X chromosome, and you have what are called trinucleotide repeats, CGG repeats, and most people carry up to about 40 to 45 of those repeats, but when they get repeated more and more, until you get to at least 200 of these repeats, the gene is silenced and doesn't make any protein, and that's been associated with the development of a neurodevelopmental disorder. And for those of you who think in terms, or are familiar with thinking in terms of standardized testing, one of the things, there's lots of things I can say about these disorders, but the thing I'm going to pick out because of the overlap is that there's a prevalent pattern where the verbal domain of functioning tends to be less severely impaired than the non-verbal domain. And so there seem to be impairments in dealing with space and time and numbers, and also what we call executive function. Another disorder I'll talk about, and I won't be talking about boys. In our study right now, we just have girls because boys tend to be more heavily affected given that they only have the one X chromosome. So this study's only dealt with boys. And all of the children I'll be talking about will be between the age of 7 and 14 years of age. Turner syndrome, most of the time, but not all of the time, is caused by the complete loss of one X chromosome, X monosomy. So you only will find it in girls because there is only one X chromosome in boys and they would not survive. So it's a population where girls have one chromosome, X chromosome missing, and it has a very similar pattern of performance when you look in these domains. There's a good deal of differences too, and I'll point those out in a little bit. But it's an interesting overlap that they deal with the same impairment. So they tend to be much, much more higher functioning than the girls with Fragile X. The disorder that really drives my lab, and it's got all kinds of complicated names, you may have heard of it if you've heard of it at all, as DiGeorge syndrome, velocardiofacial syndrome. We tend to use the term chromosome 22q11.2 deletion syndrome because it's so easy to say. Actually, that's not the reason, because it turns out that there's actually a family of disorders that were thought to be independent and turn out all to be due to this same genetic deletion of about 1.5, mostly 3 megabases of material on the long arm of chromosome 22. And again, the pattern is very, very similar. And I won't now eventually be talking about Williams syndrome. That was the original conception of the study, because we were going to study the children with Williams syndrome too. When I started these studies, I was in Philadelphia where we had a Williams syndrome clinic. But after moving here, it became somewhat difficult. But it's also true that there's a genetic deletion at 7q11.23 in Williams syndrome, and you have a very similar pattern. And these are very interesting children because they can be much more seriously affected, more heavily impaired than the other populations, but they have particular areas of incredible strength as well. All of these populations have strengths and weaknesses, but the strengths in the Williams syndrome population often to be rather more outstanding with their very hypersocial and very verbally expressive skills. We just haven't collected enough data to show you here what's going on. So what is the question I was trying to ask? Well, there's an old English joke. If you can pick it up, I actually grew up in England. And they also always say about English people that the 3 things they'll do is form a queue, drink tea, and talk about the weather. And when a whole bunch of people who studied neurodevelopmental disorders get together, they often ask this question: what is it about these different disorders that seems to be similar when they have different genetic causes? So why is it that they have this overlapping pattern? And that's really the question. Why is it that there is this overlapping pattern? What are the developmental trajectories that are apparently similar? They're not typical, but they're similar to one another. And what might this understanding do to explain how you get these impairments with space and time and numbers? And that might even tell us about how things like typical arithmetical or numerical development actually takes place. So as I've said, the question really is that there's children from several different disorders who have similar impairments, largely but not completely, in nonverbal functioning, to do with things like visuospatial, visuomotor, temporal, numerical. Very seriously with math, when these kids get to school, they really start struggling with math, and you start talking about that with the parents, and that's where the heads start nodding, and various aspects of attention. Now, people often refer to these disorders as nonverbal learning disorders. I actually really don't like that name because most of these kids really do struggle with verbal aspects of functioning too, but they do tend to be slightly more impaired, or rather more heavily impaired, in the nonverbal areas. So, if you see a child with nonverbal learning disorder or so described, much of what I'm going to say to you tonight is somewhat relevant to this. So that really works probably as some kind of a vaguely appropriate description, but what's the explanation? Can we explain it, and then might there be a way to fix the problem? So one way what we would ask that question here at the Mind Institute is there an underlying brain and mind account? Because if there is one, then we might be able to explain why this is happening, and if there is one that we can describe and explain, maybe we can figure out how to fix it. So what follows is, and I make very clear that this is not the explanation, but one possible explanation of why this might be the case. So the first question to ask is, just how similar or different really are these populations of kids? And one way to ask that question is, what would you get if you assessed them the way that most people would assess them, which is with standardized measures like IQ? And by convention, for the rest of what you'll see, with a couple of confusing exceptions, and I'll point those out to you, typically developing children will be in blue on the graphs that we have. The children with 22q deletion will be in pink. Green is Turner syndrome, and orange is the full mutation fragile X girls. As you can see, that in most of these IQ measures, this is full-scale IQ, verbal, or these are across several different tests, but the verbal domain, the non-verbal domain, and this is what's called the processing speed index. 105 or 110 is the new 100 in the typical world. But you can see there's a good deal of difference between these other 3 developmental disability populations. They're not the same at all. And in fact, all of them, to a greater or lesser extent, the 22q deletion group, very much lesser extent, show this higher verbal than nonverbal split, as it's often called. And interestingly, they all show a very big difference of at least 2 standard deviations in processing speed to the typical kids. Now, it turns out that most of the way that we do our assessments use very non-standardized experimental tests, and most of the measures that we take are very speed and accuracy related. So if this really is the case, that there's an enormous difference in processing speed, then we'd have a bit of a hard time explaining that. But it turns out that of any of you who know these kinds of tests, the processing speed measure from the WISC, for example, for kids, is very heavily dependent on visuomotor and visuospatial and executive function, the precise things that we expect these kids to be impaired at. So this is not a very good measure. So in order to try and understand what the floor might be for the kind of measures that we do that are adapted from sort of much more traditional cognitive psychology experiments, the first thing we do, and there's going to be lots of audience participation here, you don't actually have to do this test, but you can see what it's like, is we get them to do what's called a simple reaction time test, which is the simplest thing you can do in cognitive psychology. They're sitting in front of a computer, they have a button box in front of them or a microphone. Something appears on the screen. In this case, it's going to be a little alien who's going to pop out of a house, and they press a button as fast as they can. Okay, so this is what it looks like. Here's the house. There's the alien, and you press the button as fast as you can. Okay, that's all there is to it. Now, we randomize this sort of unpredictably, so you can't just press the button at a regular pace. And what happens is that when we take, and there's actually a lot of kids in the study, there's over 60 kids in these two groups and about 25 to 30 in these two groups, you find that the average response time of just pressing that button is almost identical in the typical and 22q deletion groups within a few milliseconds of one another, a few hundredths of a second, thousandths of a second actually. The Turner syndrome group is only a little bit slower, a few tens, and actually I determined that last night this is actually a real overestimate. It's more like 417, not 440. So the biggest difference we have is only about 35, 40 milliseconds. Okay? What you'll see when we look at the cognitive tasks is that the smallest differences we get are in the order of hundreds of milliseconds. Okay? So that's essentially a way of showing that there isn't a problem in just responding quickly, but in fact, as we add these tasks that are probing the specific areas of dysfunction that we think are important, we're imposing a big cognitive load on those kids. We're essentially testing a system that isn't working very well, and we can use this to essentially residualize out or take out just what the cognitive cost is. Okay? So the hypothesis is that there's a bunch of functions that depend on certain cognitive systems to do with the processing of space and time. And actually we know a lot about these systems in animals as well as in humans. They're very much, the earliest parts of them are parts of the very early mammalian brain, and that they form development of higher order functions that you learn throughout childhood, and they depend largely on eventually a network of brain systems that link the parietal and the frontal lobes, and I'll show you where those are. It's not that these systems are coded into the parietal and frontal lobes, but they come to represent those things. And that's how we get these systems to sort out. But I want to get you to think about what on earth objects and space and numbers have to do with one another. Okay? So if you think about it, space and time are tremendously abstract concepts. If I asked you how much space is in this room, it's a pretty weird question, right? It's very hard to know. Okay? They have scale. We know they have scale. They have distance or magnitude or scale. Okay? But they really don't have any actual values attached to them. But we use mental units to break them up and think about them meaningfully. So we might know how many square feet are in this room. There aren't really square feet in the room, but we can impose that value on it, okay? And what we have to do is we have to learn how much is one of these things. How much is an inch? Or how much is a mile or a second or a foot, okay? And so we learn these over time. And obviously very small children don't know what these things mean at all. They have to acquire them. And they acquire them by learning from their sense of what space and time really is like, and they impose them on the top. And in fact, as a human species, we invented numbers to talk about how much of these things we have. How many cows there are, or how far something away is. That's what numbers are for, at least in their basic function. There can be very high-level versions of that. So the question is, what if your mental units don't match the parts of the real world very well? Okay, what if you have a very poor matchup of estimates? So your space and time estimates will be very wrong, and numbers won't make sense because you won't really know what 4 means or 10 means. Okay, and I'll sort of break that up a little bit more. So if you really have, if what you think is a foot is actually 18 inches, okay, then you're going to have a hard time understanding, and you're going to have a lot more error when you try to make these judgments. The same if you think a minute is really a minute and a half long, or 50 seconds long, or 30 seconds long. If you can't match up those mental representations, And when I'm going to talk a lot about representation. What do I mean? Let me just step out of that a second. We think about cognitive mental processing really like the way that a computer works. Okay, you aren't processing the information in the world, you're processing essentially mental tokens that are a bit like sounds or pictures that are representations of things in the world, not the things themselves, just in the same way that computer software processes those representations. Okay, so the accuracy with which those are internally represented and then can be processed will determine what computations or how accurate your mental computations can be. And so if they're poor quality, then the computations will be poor quality. Okay? The best way to think about this is very often the very familiar thing. If you have a very old-fashioned digital camera, almost everyone has one now, you try and take a picture with your phone or something, and you put it up on your computer screen, and you try to zoom in and find the detail in that, very often it just turns into blur. You now can't get the computation done because the representation isn't of sufficient quality. Okay? So this is what I think explains most of these overlapping cognitive impairments. And most of the work has been done with this 22q population, but we'll see how well it translates into the others. So if we're right about this, and I think there's a good chance that we are, then if we know what's wrong, we should know how to fix it. And in fact, for the want of some, of course, what does it always come down to? Money. I'm trying to raise sufficient money to actually go ahead and try to build interventions. And for me, I think the interventions will be video games. You may not want to hear this, but video games, it's been shown, can actually improve this functioning considerably. So we want to build customized video games that will actually improve the resolution of your mental representation of space and time. And then these kids should be able to comprehend things that don't make any sense to them, and then they'll be able to learn. So we're trying to raise that money right now. So very quickly, let me just give you a sense of what we do and somewhat what we don't do very much. Most of what we do is experimental cognitive performance tasks adapted from cognitive psychology, and they're not going to be terribly familiar to you, so I'm going to make you do them. Okay, then you'll know what it's like. And we focus on things like attention and, go back a moment, attention, enumeration, magnitude judgment, because these higher level things are built on these lower level things, because attention is the way that we break up space. What should we attend to and where should we attend to it? We do a lot of MRI analysis, looking at the structure of the brain, the connectivity of the brain, and the function of the brain, and all of these things don't make sense standing on their own, but they're related to one another. And we do standardized neuropsychological testing, things like the WISC, for general intellectual function and academic testing, achievement, the key math for looking actually at numerical functioning, some executive function things, the judgment of line orientation, all kinds of things trying to get at this in a more broad sense. But the action really comes from here and from here. So I talked about frontal and parietal areas of the brain. This is the consequence, not the cause of the problem, I believe. But this is where most of us end up representing and processing these kinds of information of space and time. There's a lot of subcortical structures deep in the middle of the brain. But if any of you know someone who's had a stroke, which very often affects the parietal lobes, very often people will lose their entire sense or a large part of their sense of space and time, and sometimes numbers, because most people have developed those systems within these parts of the brain. So here is a brain. Unfortunately for us neuroscientists, they don't come colored like this. It would help if they did. Here's the occipital lobe, mostly visual. The parietal lobe, we're going to talk a lot about the temporal lobes and the frontal cortex. And mostly what we're going to be talking about is systems that are dealing with the posterior parietal lobe, both the inferior and superior portions of it, and also the prefrontal cortex. Okay, so when I talk about frontoparietal networks, I'm talking about the relationship between these two areas that are formed in networks together. We don't think about a piece of the brain does this particular thing. We have networks that organize things. There's actually a piece up here called the frontal eye fields that's very associated with actually moving your eyes or picking up visual stuff, and you'll see that coming up later. So what's the hypothesis here? My hypothesis is, I've kind of told you something about already, that spatial and numerical and to some extent executive impairments derive from these lower level impairments in these building block functions of space and time that we learn about very early on, and that typically, so in the typically developing brain, come to be implemented by these frontoparietal networks. And if you take typically developing adults and you put them in an MRI scanner, and you ask them to do this, you'll find that the posterior parietal lobe is very involved with representations of space and time and number. And when you get damaged that, you very often find that goes awry. And the prefrontal cortex has a lot to do with working memory and cognitive control, very often the control of attention. So when people think of attention, they often think about attention deficit disorder. And that's more this kind of a system. Whereas here, it's more about moving your attention around in space, like having a flashlight in a dark place where you select information. Okay? And so the idea, well, our first hypothesis is that these these mental pictures which have lower resolution create errors in the system because you can't represent and process them very well. So like I said, it's just like trying to compare some detail. Which of these two small things are bigger than the other or further away from the other when one of us has a 1 megapixel digital camera and one has an 8 megapixel digital camera, and we take the same picture, but when we zoom in, this poor person really can't get there and this one really can. So as we think about space and time, my hypothesis is that these lower resolution images or mental representations don't allow you to think with the same degree of clarity and learn about what these concepts mean. Okay? The other thing is that there are neural network connection differences that affect at least speed and maybe more general functioning because different parts of the brain are not coordinating with one another properly, but very much at least speed. And we found that we think the cause of this is problems in the lower level subcortical areas, but eventually it hampers the typical development of these neural networks that in most typically developed adults will run these systems. So think about it this way. You can drive from Sacramento to San Diego on I-5, or you can go down 101. Okay, you'll still get there if you get down 101. But you know, it's going to take a heck of a long time. You're going to be a whole lot more tired, and if you had things to do on the way or when you got there, you might not get to do all of them. Okay, so you can get the job done, but nowhere near as well as you'd like to get it done. And so all of these following experiments I'm going to talk about are designed to test these hypotheses. Now, I always want people to see and feel what it's like, okay? So on the next slide, I'm going to show you an image. And over about here, there's going to be a cross. And what I want you to do is fix your eyes on this cross and don't move them. Okay, you promise? Okay, so look over here and take a look at that cross and don't move your eyes. You doing it? Okay, now without moving your eyes, tell me if you notice anything on the other side of the screen. Some bars. And would anyone like to tell me, without moving your eyes, how many there are? 4. Most people are saying 4. Okay, you can look now. Okay, what I just gave you was the ability to see what it feels like to have reduced resolution in your attention. Okay, because we know that attention is like vision. I'm not saying this is a problem with vision. I'm saying this is really a problem with attention. And attention gets to be lower resolution out of the edges, just like vision does. Okay? You can't individuate these things because they crowd one another and they fight with one another, okay? And so you really can't tell. And so what if this was happening much more towards the middle? And notice how you undercounted, okay? And that's what we see a lot. So let's do one more, okay? Actually 2 more. So now we're going to have a cross up here. I want you to take a look at this cross up here, okay? Don't move your eyes. Are you aware of the circle on the right? Anybody know what's in it? Huh? A C, pretty close, take a look. Letter G, so it was not easy to get a hold of it, but you know there's a letter G. Now let's try one more, so fix your eyes over here, and now let's see, you can tell me what's in there. Pretty hard, huh? It's exactly the same G, but now what I did was I crowded it with some other things. Okay? And now what happened is your attention really got kind of lost because I was crowding things together. So that's the sense of what's going on here. That's like having a lower resolution because these things are merging into one another. Okay? And it's making it very hard to disambiguate them. And crowding is very important. So it's really a bit like operating in a world where, these are kind of crummy pictures, but I have a ruler like this, and somebody with this disorder has a ruler like this. They can still make a measurement, but it's going to be nowhere near as accurate. They're going to be missing, so they're going to make sort of more error. And, somebody might have this clock and somebody might have this clock. So you know that's, 10 past or 10 till, but you really can't tell between 10 past and 20 past. Okay, so it's reducing the resolution of that. So how do we know this is what is going on? Why do we think this is what's going on? So I'm going to show you a whole bunch of experiments that, and I could go on and on and on all night, but I don't want to bore you to death. But I'm going to have you do some of them. Now notice, most of these experiments are done with 7 to 14-year-old kids, so they're kind of very kid-friendly. So if you're feeling tired at the end of a long day, and I know I am, then these should help you. So here's one where you have to measure parts of space, essentially. And this is actually adapted from a test of people who have had a stroke. And your task is to tell, in the real situation, you have a little button box, you're sitting in front of a computer, you have to press a button, a right button or a left button or one in the middle, and you'll see that you have Kermit the Frog in the middle of this line, and Fozzie Bear and Miss Piggy are at either end. And you have to say which side, who does Fozzie Bear like more, okay? Now here he's right in the middle, right? Who is he closer to this time? Okay, that's easy. And this one? A bit more uncertainty. Actually, he's ever so slightly close to Miss Piggy, okay? What happens if you do this? So when he's close to one end or the other, or right in the center, it's very easy. But when you get away from that center and you're not anchored in one of those places, error goes up, and you could tell your own uncertainty probably, okay? So we get this very typical M-shaped response. This is one of the few places where the colors I told you about don't fit. So here the 22q deletion kids are in pink and the controls are in yellow. So here is basically that distance. Here's the middle of the line, here's the edge. And you notice that no one makes any errors at all. This is the degree of error. No one makes any errors at all at either end, and it's a very low error right in the middle. Neurologists like to call this the zone of indifference. Okay, you're not really sure. And since the right hemisphere tends to dominate left space, you get a little bit better on the left and a bit worse on the right because it's a little bit harder on the right-hand side. But you can see that there's actually an increase in the error that the 22q deletion kids have. And if there's one slogan in my lab, which we came up with years ago, is the difference has to be bigger before you can see the difference. And we just go on and on and on. So if you're teaching kids, or you have kids who have these problems, think about that. The difference must be bigger before we can even see there's a difference. And that's a big, important piece of this. So when things are crowded together, clumped together, just difficult to disambiguate, they're just not going to be seen that they're actually different, okay? So what happens is, in fact, the resolution of these mental images is about 30% lower in the kids with a deletion. And in fact, a colleague of mine in Switzerland, with a slightly different group of kids, they call it VCFS, for velocardiofacial syndrome, did almost exactly the same experiment with time, okay? So you hear or you see something for a period of time, a tone, like whoop, that goes away, and then you get another one, and you have to say, was the second one longer or shorter than the first one? And you get them closer and further away until you figure out where the threshold is. And here, this is their typical kids, this is the general distribution, this is the median response, the median accuracy, or basically how much difference there had to be. And in both cases, the auditory and the visual case, the kids who had the deletion have a much bigger threshold. Again, they need a much bigger difference before they notice there was a difference, okay? The temporal resolution is about 35% reduced, okay? So, there's a lot of evidence then already that, that there's problems with space and time. And it turns out that when we think about quantities, we often think about them very abstractly. And when I mean abstractly, we tend to think about them very spatially, okay? If I ask you to do a very weird thing for me, would you point to where in space the number 1 is? Okay, where's the number of 100,000? Right, generally out this way, right? So we tend to think left to right because we learn to read left to right. Small numbers are here and big numbers are there. And in fact, the bigger they are, the further away from one another they seem to be, okay? And when you think abstractly about numbers, we often think about that way. So what happens is, and you can show this in animals too, is that adults create this analog representation of approximate quantity. And in fact, if you have them do these kinds of tasks in a functional imaging experiment, they show a large network of activity with the posterior parietal lobes and that frontal lobe. And near quantities, things that are closer to one another, much harder to disambiguate than ones that are much further apart. It only does it by a couple of milliseconds, 10, 20 milliseconds, maybe 100 milliseconds, but you can see a difference, whereas it's really easy to tell the difference apart. They do it very quickly. And even infants show this phenomenon, so do animals too. So if you get infants to discriminate sets of objects, there's something called the Weber fraction, which basically means that bigger differences have to be bigger before you see them. Look, there it is. It's actually a law in physics, the Weber's law. Okay, so the prediction is that this reduced resolution will have the biggest effect when the difference is the smallest, okay? Because it's got to be bigger before you see the difference. And in fact, so the other prediction is that to the extent that there's disconnectivity, and when I use disconnectivity with a Y, which means it's not that one thing is not connected to the other, it's poorly connected, okay? Like dysfunction or dyslexia. So that should at least impair speed and maybe general function. Okay, so here's another task you have to do. You have to pick which one of these two things is the bigger. That's pretty easy. Or which one of these two things is the bigger. Now, they're the same size, but they're not conceptually the same size. Okay? And we vary the difference or the distance, the numerical distance between them. Okay? And it's much easier to confuse them the closer they get. And one of the reasons is, as I mentioned, that people tend to represent quantities in this way. So small numbers tend to be squished together and large numbers tend to be further apart and they tend to be spread in this direction. So smaller distance between the two things means they're less distinct. Okay? So the resolution should affect the small but not the large differences. Okay, now we get to our color scheme. So what you're going to see generally along this axis here is a blended measure of time. What it's measured is time and accuracy combined. Okay, so how long did it take you? And if you make more errors, you actually get a cost to your response. It slows you down. That's because we want to actually blend in accuracy and speed at the same time because we expect these kids to be doing more poorly. And these are the differences. So these are actually, in the case of those bars, they're 1 centimeter differences. Okay, and so what you see out here is that there's large differences, 5, 6, or 7 centimeters between the two bars. Notice, remember we said that the biggest difference was about 40 milliseconds between these groups? Now, this is 600 milliseconds, this is nearly 750 milliseconds. Even out here, so the intercept is enormously different. We've doubled or tripled, and in fact with these two groups there was no difference whatsoever. So there's a real cost even when they aren't actually doing a whole lot worse out here. But here the slope, see, notice how the small differences are kind of impair the typical kids, and then they balance off pretty quickly. But all of the disability groups have a great big slope. They don't really settle down to here, and even here they're not terribly settled down. So they need a much bigger difference before they actually can perform the same as the typically developing kids. And that's also true when you deal with numbers. Now the numbers are a little different because they're more semantic knowledge, okay? So you get kind of a more linear degradation in the typical kids. And one thing to notice is this is sort of a weird phenomenon. When you test kids, occasionally one child will change the profile slightly. But in general, we've seen that the Fragile X and the 22q deletion group are pretty much identical, and apart from this one thing they are. But notice how the slope of this line is very much more like the slope of this line than these are, although it's incredibly much slower. Okay, even though there was only about 20 or 30 milliseconds difference. And that's because I believe the Turner syndrome kids are much more high functioning. We know that they're much more high functioning. So they've actually got a whole lot more conceptual or semantic information, and they can kind of improve a little bit compared to where they were when they were just having to do it on pure spatial representation. So because they're more high functioning, they kind of conferred an advantage in that sense. Okay, so there's another example of resolution and how you get a handle on thinking about quantities. Now, I talked about attention. So let's think about attention. Everyone, as I said, mostly thinks about attention as attention deficit disorder. That's the front part of the system. So if the world were dark and you had a flashlight, you'd be moving it around. Only what your flashlight lit up is what your brain is processing. Okay? That's what we call spatial or attentional selection. And that's driven by this parietal part of the brain very heavily. When you decide what it is you want to pay attention to, you have to decide not to pay attention to everything else, right? And that's the more frontal part of the system that tends to be more related to ADD. So it turns out that you use attention to quantify. Okay, if you have a little kid, and I have one at home, and he's trying to count, he very often completely messes up because he keeps counting, goes over things several times, but he wants to touch everything because he can't control his attention very well, so he gets a tactile response. But we learn later to do it in an easier way, and I'll show you how that goes. So if you have very small quantities, 1 or 2 or 3, they tend to jump right out at you, and there's a made-up word called subitizing, which means they're automatically apprehending. And we know that that doesn't require spatial attention, shifting attention around to get that to happen, and you'll see it in a moment. Some work I did with a colleague in the late 90s showed that that's true. So we don't use that spatial attention system, and therefore we don't really predict there will be an impairment because there's no resolution issue here and there's no attentional load, okay? However, when you get to quantifying large sets, you do have to use a spatial attention system. You do have to move it around and control it. And it's very heavily involving that frontoparietal system. So that's where we think that you'll get impairment. It'll slow down because of that disconnectivity, and the resolution is going to ding you on that situation. So you ready to do it? Okay, so what I'm going to do is I'm going to show you, oh, let's go back a second. This is kind of easy. That should have popped up there. So what I'm going to do is I'm going to show you these green squares on a red background, and I want you to yell out as quickly as you can how many green squares there are. You ready? That's pretty easy. Ready for the next one? Okay, there was a pause there. Did you notice that? As I like to say, that's the sound of attention being shifted, that silence. Because probably what you did was you went something like 1, 2, 3, 4, 5, 6, 7, right? Or you got a bunch of them together. You grab that bunch and you grab that bunch. So you're sort of using this strategy for that strategy. But you had to sort of navigate in space. And notice, if in fact you really are having a resolution problem, these guys might actually bunch together and you might lose the difference between them. Okay? So, what do we find? When there's small numbers, there's no difference at all between the groups, in speed or in accuracy, for the first 2 or 3 items. But as soon as you get out from that point and you have to use spatial attention, whammo. First of all, the speed goes slower and slower and slower, and errors go up and up and up, okay? Because you can't control your attention properly and you're running into this resolution problem. And here's a really interesting thing. I just learned this afternoon because we just got some brand new data for analyzing these other groups. Because these other groups, the 22q group, we've known this a long time. The other groups, we've just gotten to the right kinds of numbers. It turns out that when the kids in the typical group make an error, they only make errors out here. Almost all the errors they make are +1 or -1. Okay? So they say, okay, you know, it's one bigger or one smaller. And they do that 50-50. But these guys all make errors 70 or 80% of the time. What do you want to guess? Too few, exactly, because they're kind of losing the resolution. Most of their errors are undercounts, okay, because they're losing them. Okay, so just real quickly, I want to bang through a couple more. So here's a task where you look at other aspects of attention. So you'll see one of these 3 displays. You'll never see all of them at one time, these little guys in spaceships. And you have to press a button to indicate which one, either the one by himself or the one just in the middle, which direction he's pointing. We have all kind of cover stories for these things. So here, they're all telling you the same information. Here, they're actually confounding one another. So you have to inhibit this information. So inhibiting, these guys are called flankers. Inhibiting this irrelevant information is called is inhibition, and that requires sort of frontal executive attention. No problem here, and here there aren't any flankers. And so what we do is we do several kinds of things. So sometimes there are no cues at all. So this is what the screen looks like. There's a central fixation point. Sometimes the target just pops right up here. No warning at all. Comparing the times when there's no warning to one where there's a warning right in the middle, then you actually get what's called the alerting system. Were you ready to accept the information? That's one kind of attention. Sometimes there's a spatial cue that's valid. In other words, this thing will flash and it draws your attention here, and that's where the target shows up. Okay, and that helps you. Sometimes there's an invalid cue. It draws your attention down here and you have to shift back from there, and that's obviously a big cost. And sometimes what you get is a neutral cue. It's right in the middle, and that doesn't help you at all, and so you really don't know where to go, and that's kind of variable, and people use different strategies. So we can look at the alerting system by comparing were you ready. We can look at the spatial selection system by looking at how these spatial cues affect you, and we can look at the executive system by looking at what happens when you add these flankers or you take these, not so much add them or take them away, but when they have the same information or the confounding information. So here is a lot of data from these same groups. So here's the spatial task. Here's the valid cue. Everyone does better in the valid situation because we just told you where to put your attention. And then usually what happens is the neutral and the invalid situations are pretty similar, statistically not different from one another, sometimes a little bit worse. And we've actually found in other tasks, this is a very subtle and complicated set of experiments, this particular variant, you don't see a greater slope. In other words, these aren't terribly different from one another, and it doesn't get harder and harder here, although it does seem to be affecting the Fragile X group a little bit more. In other versions of the task where we manipulate other things, we generally find that it gets worse between the neutral and invalid. And those require slightly different neural systems. But let's not belabor that point. What you can see is that very small reaction time difference turns into a very large difference that's pretty similar for all of the groups. So they seem to be similarly impaired on spatial attention. They seem to be very similarly impaired on the alerting system, though oddly enough, you're supposed to respond more slowly when there's no cue to one when there's a neutral cue. So actually, some of these guys are going backwards, and we can't quite figure out why that is. But the differences are so small, it basically really means that they aren't really showing a difference at all. But there really is a difference here in the Turner syndrome group and the Fragile X group. There's a big impairment to not being given a cue. It's like, oh, I wasn't ready. They're not getting the information. And then lastly, when we have the incongruent flanker. So here you have all the guys going in the same direction. Here you have them going in the wrong direction. Of course, it slows everybody down. But look how much steeper the slopes are for most of these groups. They take a bigger hit on all of those things. So generally, there's a lot more cost in all areas of attention for these groups. Now, here's the question. I'm going to try to get through this quickly, but without confusing you completely. I want to look at what the neural basis of this is. Is there something in the brain? I'm going to pick out one small thing. But I'm going to teach you a little bit about diffusion tensor MRI imaging. You ready? In about 30 seconds. What is diffusion tensor imaging? Diffusion tensor imaging is a way that we can image connectivity in the brain. Not structure, not where things are or how much stuff there is, but what's connected to what. And it does this. So there are a few technical terms here, but don't get too frightened because I'll simplify them. What we can measure is basically three main directions. It's really X, Y, and Z, okay? Three major directions, and we're going to get all the little subtleties in between them, but those three major directions are separated in a particular way. The first direction is called the eigenvector in the technical language, but basically what it means is, so here's the thing. In your brain, you all heard the difference between white matter and gray matter. Is that familiar to everybody? Gray matter is the neurons in the brain, and white matter is sort of the protective stuff that goes around the connections. So it's really like when you look at wiring in a computer system, you've got all these wires going on, there's plastic wrapped around them for insulation. That's how the signal gets through. So if you lie lots of cables in the same direction, there'll be lots and lots of that protective coating all going along that fiber tract. And it's got lots and lots of fluid in it. Okay, there's lots of fluid in your brain all over the place. Some of it's completely unconstrained. It's in a ventricle where there's supposed to be space filled with fluid. And so that's very easy to pick up because when you shake it all up, it goes in every single direction. But when you have this fluid that's trapped in that white matter, it's trapped along the direction of the white matter where the fiber tracts are going. So you can actually pick that up, and I'll show you how we do that. So the first direction is in the parallel to the fiber pathways. In other words, it's the diffusion of water that goes along the axis. Okay, so if everything's going this way, it'll be going in that direction. The other two are the two main other directions. So if this is X, this is Y and Z, okay, and they're perpendicular. So if you average those together, you just get everything that's not in that main direction. So you can see things that are going the other way, okay? And there's this magical thing, which most people don't know how to say, fractional anisotropy. So isotropic means all in one direction, iso and tropic, one direction. Anisotropic is not in the same direction. Okay, so I just want to, if you ever hear that term ever in the future, you'll know what it means. So let's just show you what I mean here. Imagine you took a glass of water and you looked at a particular molecule of water in that glass of water. The molecules would just be flying in every imaginable direction. And so you could average them out and eventually they would look like a kind of a circular soccer ball if you gave them enough time. Okay, they're unconstrained. But if you take some fluid that's trapped in white matter that's along a fiber path, so here's some slices of the brain, and here's what we call the internal capsule, where there's very oriented fibers in the internal capsule. And you looked at those, because they're trapped in the fibers, they can't go in every direction. They're sort of more constrained along this primary axis, okay? And that's what we call the axial direction, okay? And so when you image the brain, particularly with certain kind of images, here's a slice of the brain. This is an image that's done so that we can emphasize fluid. Here's brain tissue. The bright white is the fluid. So those are the ventricles in the middle, big empty spaces where there's just literally CSF, cerebrospinal fluid. And if you shook all the molecules up, they'd go in every direction because there's nothing constraining them. And then here's the rest of the brain. Okay, can't see it very well. This is what's called the apparent diffusion coefficient. All that means is how much diffusion is there. So there's most diffusion in that ventricle because if you shake it all up, there'll be a lot of motion there. But if you go back to that anisotropy thing, which is how much orientation is there, okay, then you see the opposite of that. So now there's almost none in here because there's nothing constraining that fluid. But here are the main fiber tracts in the brain between the two sides, the corpus callosum and front to back. And there's also ones going up and down in this plane. So here's front, back, up, down, and left to right. Very hard to see until you color code them. But now you can actually capture this. So here's the corpus callosum at the front, going left to right. Here's the corpus callosum at the back. Here's the longitudinal fasciculi connecting the front and the back of the brain. And then going through this plane of the thing are the corticospinal tracts. So we can actually build wiring diagrams of the brain using these. It's very cool. Okay. So given that we can do that, we discovered something kind of amazing, which is when we looked at the 22q deletion brains, we found, without looking in a specific place, that there were actually 4 clusters where we got different signal, where essentially the opposite signal was going on, okay? So in these 4 clusters, the 22q deletion kids, essentially, I'll oversimplify this so it just makes it pretty obvious. Can you see that there's sort of the white tracks along here? Are those pretty obvious from where you can see? These are the main tracks that are connecting the front to the back of the brain. And you can see the gray areas right next to them. That's the cortex, okay? So you would expect most of the diffusion to be going along this direction because that's where the fibers have been laid, okay? And what we found is the 22q deletion kids had very high orientation in this direction. Whereas in exactly the same places, because we pulled them out because they overlap, the exact opposite was true. The typical kids had high perpendicular orientation, which meant that most of it was going away from that direction, and of course that assumes it's connecting to the cortex nearby. Now these four areas are classical areas in that frontoparietal network, okay? Suggesting that it's like if this is a freeway, these guys, the 22q deletion kids, appear to have lost all the exits to the cortex nearby that they should be trying to talk to. It's like pulling all the plugs out. Okay, or not all of them, but many of them. Whereas the guys who have very high radial diffusion have lots of connections, lots of freeway. So they can talk to that cortex pretty well. And we found not only that difference, but when we correlated it with the performance on one of those spatial attention tasks, it was incredibly significant and in opposite directions. So the more of that backwards and forward diffusion the 22q deletion had, the worse they did on the attention task. And the more of the other kind of diffusion the other guys had, because they presumably were talking to the cortex well, the better they did on the task. Okay? So now we've found some real neural changes that seem to really relate to performance on one of those critical tasks. And here's the amazing thing. We have a little less data so far. It's in the process of being analyzed now. Turns out that everybody seems to show that signal. Okay, here's a kind of pretty low resolution picture of the brain, but we did it so we could cut it open. So here is that same location. Okay? Now, the colors got changed again, weirdly. The Fragile X kids are in red. The 22q deletion kids are in red. There's a difference from typical. The Fragile X kids are in yellow, a difference from typical. And the Turner syndrome kids are in blue. And in fact, you get green here because you've got red and green and blue. Okay? And so if we look at it in terms of the actual brain, this is essentially, if we took all the brain tissue away and we just left the fiber tracks and we're looking down from above, this is the front of the brain, this is the back of the brain, and we're going from low down in the brain to high up in the brain. We go on through, and here you see the corpus callosum, and in the 22q deletion kids, we see that signal on both sides, and when we get to the right area of the inferior parietal lobe, you see that all three groups have the same place where they're different from typical kids. And I would lay money on the fact, though maybe I'm dicing with death here, but when the numbers get bigger, I think we'll see it on both sides in all of the groups. We just haven't got that yet. So that's one way to look at it, and here's another way to look at it. There it all is on that one side, and here it is on both sides. Just looking from the side. There's the corpus callosum, front of the brain, back of the brain. Okay? So really quickly, I've got one more task for you to do. Okay, what I want you to do here is I want you to look at these planets that are flying around and just watch them for a moment. Okay? So here you are lying in the fMRI scanner, and you're looking at these on the screen above you. And you don't have to do anything, you just have to look at them. Now in a moment, they're going to stop, and you'll see three little green aliens hide behind three planets. The planets will flash. Here they go. You've got to follow them. Don't lose those three. Here we go. Seems very hard to do, but let's see. In a moment, they'll stop, and a question mark will come up, and you just have to tell me, did that planet hide one of the aliens or not? See? Not so difficult after all. But it's a pretty taxing task, right? So that's a pretty hard condition. We actually do much simpler ones with the kids. So what that does is that turns on, we now know from a lot of adult imaging, this really turns on that spatial temporal attention system in the brain, that frontoparietal network I was talking about. It just blazes when we do this, okay? So what happens when we look at the kids in these different disorders? Okay, so here's what happens. This is kind of blurry because we've got a lot smushed together, but I want to sort of show you what I mean. So here we have a bunch of kids, and by the way, my amazing postdoc, Elliot Beaton, produced this latest image today. So this is right hot off the presses, because we finally we introduced the task very late with the Fragile X girls, and we finally found a girl who was high enough functioning and was able to stay still enough that she actually performed at the same level of accuracy. It's actually important that you perform at the same level as accuracy, otherwise it's hard to interpret it when you do a task like this, but that's a technical point. So anyway, these are very conservatively imaged. Here's the front of your brain, here's the back of your brain. We're looking at it from sort of the top, as if you're going back down like this, looking at it from one side and the other side. And here, let me see, the colors keep getting changed. In the typical, so you see kind of green under here, that's that parietal area where you'd really expect the brain to be activating. And here's the frontal eye fields. Wait a minute, I've got to come around here. Okay, so let's just look this way. Yeah, so here's the parietal areas, here's the frontal eye fields. Here you see the frontal eye fields and the parietal areas right here. So that green is exactly what you see with typically developed adults. That's the canonical, what we call the typical network for this task. You always see that kind of thing when you use typical people in this task. Now, if you look at the 22q deletion kids, you'll see that there's a lot of mushy red stuff that actually, some of it overlaps exactly with the green in both places. When you get brown, you kind of get some mixture of that. But they also seem to be activating a lot of other brain areas, okay? You look at the Turner syndrome girls, there's 7 of those, and you see in green, in blue, then you also see here that they're activating, again, very much the same thing, but also a lot of other areas too, but not terribly different ones from the 22q deletion kids, but still some similar kind of areas. And then finally today we're able to add the one girl with Fragile X syndrome. You really can't hardly even see her because she's in orange, but she's actually kind of buried under here, and she's producing, actually, she's a very high functioning kid, so she's probably more like a typical kid, actually. And so she's producing a very similar network, but again, slightly different. So what's interesting is that it turns out that we really seem to be showing that it's not that they're doing something completely weird with their brain, but they're actually kind of cranking away hard at this. And this is only one target. They're all doing it at 90% correct, so they're all doing the task the same way. But they can't quite use the same brain network. Now, I want to be very clear about this. Because they're not using the same brain network as the typical kids, doesn't mean to say they're using the wrong network. The right thing way to think about this, when you have an atypical development, it doesn't mean that they should be using the same system, and if they're not, it's broken. They've developed a different neural solution to the world because they started from a different place, right? And it's probably not the optimal one. So one of our big questions is, if we can intervene, if we can change functioning, because the difference between the computer model, where you've got hardware and software in the brain, that you can't change a computer by changing the software, it doesn't change its wiring, but that is true of the human brain, that you get changes in white matter and connectivity all the way through your life, particularly into early adulthood. So make the brain do things differently and it'll actually structure itself somewhat differently. So if we can intervene and we can change things, we might actually either push this network back towards the typical one, or in fact get this network to be more efficient so it's actually doing it even better, and we can actually reduce that difficulty in the resolution is what I think would probably go on. So I really think there's the possibility of intervention. So let me just wrap this up then. There seems to be a strikingly similar cognitive endo phenotype. And I wanted to introduce this big cognitive, this technical term, because you probably heard of genotype and phenotype. You hear that all the time. The phenotype is the manifestation of the genotype. And endo phenotype means it's somewhere in between the two. Something you can measure much more accurately between the immediate original cause, which is some change in genetics, and the outcome, which are these impairments. So endophenotyping, finding a way to sort of describe a medium point where you can quantify things very well, is a lot of what we do. Okay, so I thought it'd be kind of interesting to use that term. And they have this almost similar endophenotype in mind and in brain, even though the standardized measures show these groups to be quite different from one another. Sometimes they're differing on 10 or more points on an IQ score, which is almost a standard deviation. Okay, that's a big difference. So it does suggest that there is reduced resolution that's affecting essentially the cognitive software. In other words, the representations and the processes across at least 3 different disorders in a similar way. And there seems to be some preliminary evidence of some changes in connectivity. And that seems to be something to do with the fiber tract integrity or the networking that's been set up in the neural hardware, if you like. And so this may be some evidence of what we call a common neurogenetic pathway. You start off in different places, but you converge into a same pathway because maybe we have a vulnerable system here that could be knocked off in a range of different ways. And if that's true, we might get developmental convergence on a similar end state, which is the description I'm giving you, essentially this endophenotype, starting from different start states. Okay. Well, so what? Well, the whole point is that if we can really show this account is right, then the advances in our research give us a better understanding of what the software is that's running in the minds of these kids. In other words, how they're processing the information. What is the nature of the stuff that they're processing? I call this hypergranularity, because it's like having an image on your screen that's much more granular or grainy because it's lower resolution. okay? And also looking at how the hardware has changed, because the change in the hardware will change the way that software runs and also the nature of the data, okay? If we can explain it, well, then maybe we can fix it, okay? So think about taking your car to the car mechanic. You can give them a big elaborate description of what it is and what it isn't doing, but because they know how it works, and that's the critical 3 words, how it works, they can say, well, I'm going to test this system, and if it comes out that way, I'll do this. When I'm going to test that system, it'll be that thing. So if your car isn't starting, it could be the fuel system is broken, it could be your battery's flat, it could be all kinds of different things, and you'd have a different solution, but you need to know how it works so you can test the system and how to intervene with it. So it does seem that there really is plasticity or change in the typical spatiotemporal system. It does get better and sharper, in other words, higher, more highly resolved with practice and stimulation. And this data comes from a bunch of college kids playing these first-person shooter video games you don't want your kids to play, right? But if we take the engines for those games, and they're publicly available, and basically make new versions of the games that are just going to respond when you get better at your speed and your spatial distribution, I think we can improve this and change the brain as well as changing the mind. And therefore, if together with families and our own work, we can get together to do this research, develop the funding to get these new novel things done, we can test out to see if we're right, and if we are right, we can build interventions and actually hopefully make changes. And we aren't going to change the outcome, we're just going to change the readiness of the brain to learn that stuff. So it still comes down to the families and the teachers and everybody else to get it done, but we're preparing a much better prepared system that understands what it is that we're telling them, okay? These are the people that really did all of the work. I'm just telling you about it. And we have an amazing group of people here. And I want to thank all of them. But most of all, the people that critical to thank are the families that participate in our research, because without them, we'd be sitting staring at the walls. Okay, thanks. Any questions? Yeah.
All your studies have been done on numbers and spaces and sizes. How does that show up in verbal or logical response? Is there the same time difference?
Did everybody hear the question? So it doesn't. I mean, the point is that there are some places where these things overlap a little bit, but it's you know, it really is that these are the critical impairments that these kids are suffering from. These are the impairments that are much more in the nonverbal domain. And so so you have to be very careful. You can't infer that there are things where you really haven't got a similar basis. This is what we very, very much call evidence-based sort of research here. You've got to sort of get to what the explanation really is, and you can't really infer to something that shares a very different basis, like a different piece of software. If you understand how a word processing software package works, you wouldn't be able to explain how a statistical package works, okay? However, having said that, there are some things that are similar. So one of the things that you run into with problems when you run into issues of space and time and things like that is you run into problems with sequences, okay? So learning what logically comes after something else is a problem because you can't really tell, it's hard to learn before and after. And even spatial language, okay? What is before and after? What does bigger and smaller mean? What is, above and below? When you're spatially challenged, those things can be a problem. So it actually can leak into those things pretty effectively, but we're really trying to explain, we're trying to very much focus on these specific functions because those things we can explain without loosening or weakening our explanation. And therefore the same with intervention. We just wouldn't say it's going to solve everything, it's just going to solve these things if it works. Any other questions?
You say that finding another task for them to do while they're trying to do, I teach an ED class at the school district, high school level, and like teaching algebra, and I have ADD, ADHD, bipolar, schizophrenia, you name it, I've got the whole gamut in there. And I'm finding my kids with ADD, ADHD, as far as math, they really struggle with it. And one day, purely by accident, we had what was a late start, and they could come in early and work with me if they wanted to. And they all had their little iPods and stuff, and I said, okay, you can listen to them if they're down to where I can't hear them. It was amazing the output in the math functions that I got from them listening, and it wasn't classical music. It was rap, it was Ted Baker music, it was but something that I wouldn't be able to concentrate to myself. It really kicked them in at a different level.
You're saying that they did better when they were listening to the music?
Yeah.
Okay, so let me make a couple of points. What I'm explaining here is not a story of ADHD, okay, but I can make some inferences and some interpolations. So I went back, let's go back to that notion I said, whereby you have a flashlight in a dark world, okay? That's what selective attention is. So only what your brain processes is what your flashlight lights up. That's what selective attention is all about. Now the flip side of selective attention is inhibitory function. When I decide to focus, you're looking at me, okay? There's something up on the wall over there. It's not within your zone of attention, although it is in the very periphery. If you wanted to focus on that and have your brain process it, what would you have to do? Disengage your attention from me and put it over there, okay? Now, because you're able to inhibit that information and keep your flashlight focused here, you can process the information, which means you can actually do something sensible with it using the brain power that you have and the knowledge you have. Now, what if we just start moving our flashlight all around the place, okay? Then nothing is going to get really processed in a consistent way over time and space, and we can't bring our knowledge to bear on it because it keeps going out of our focus of attention. We actually use the fact that many of these kids have attention deficits, because you really can't disturb one part of the system without disturbing the other part of the system, as a very important way that we get. So how do you get a kid in an MRI scanner? Okay, you would think this would be an insane thing to do. They're anxious and they're impaired and they're hyperactive some of the time. Not really hyperactive, but they're distracted. And one of the ways we do it is, I learned this very important thing a long time ago. Martha Denkler, who's a tremendous pediatric neurologist, used to say that, and I think she probably still does, that ADHD is not a distractor phenomenon. It's an attractor phenomenon. I'm attracted to this, no this, no this, no this, no this, no this. And I can't inhibit it. So I can't process that information. So I think what's probably going on in your case is when you gave those kids something familiar and comfortable, they just shut down and they went into their bubble. And now they were able to focus on what they were doing, as opposed to being focused on all these other kind of things. And the math just goes, whoof, straight out the window. And so what we do in the scanner is we give kids a movie to watch when they're not doing the functional imaging task. And you know what kids are like when you put a movie in front of them. You keep very still that way, because that's the most interesting thing in the world. And they pick the movie. So I think that's part of it. So this is not a story about ADHD, but ADHD has a lot to do with not using your selective attention well because of this link between inhibitory and selective attention. So you probably gave them a way to essentially take the outside world away and allow them to focus more. As for all the others, that's a whole other challenge. Yeah.
Can you say one quick word about if there are therapies already developed, how effective they are, and is it a permanent improvement?
No, I can't say any of those things, but I'll tell you what I think is going to happen. So the fact is, let me just make 2 points. One is that there are a lot of very skilled clinicians and therapists out there. And if you have a child who has nonverbal learning disorder, if that's the term that tends to get used, there are a lot of people that know a lot about how to help them, okay? Lots of skilled clinicians and therapists can do that. But they're doing on the basis of what has worked in the past and what they think is going on. I often use the differences. So what I'm trying to do is, imagine you have, I'm not meaning to disparage anybody at all, I'm just trying to make a contrast here. You can have a therapy that's rather generalized. So you could take a multivitamin every day, okay? And it improves your health in general and your immune system, and you may not quite get sick, okay? But if you have a particular virus, and you can take a drug that goes right at that virus and kills it, then that's going to be a much better solution, right? So what I'm trying to do is essentially do what's akin to a drug development project here, which is figure out what the receptors are in the mind and the brain for this problem. We know what the delivery vehicle is like to be. It's video games, because it's been shown to do that. Now we have to figure out how to get that delivery vehicle to talk to those receptors by optimizing the game and then seeing if it works. So we haven't done it yet, but I really think we can do that. And there's a lot of evidence out there that it might be very effective. It's not going to solve the problem, as I said. It's going to reduce the problem with the resolution so it gets the brain ready to learn rather than fixing the problem all the way through. As to whether it will last forever and how expanded the effect will be, it's unclear. My prediction would be that it would be broadly applicable, because a lot of things depend on that. So all the things that depend on that, you'll be able to learn better if you spend the time learning them. If you just wait for it to happen, it isn't going to happen. But that's the same true of development. If you have a child and you lock them in a dark room and come out 20 years later, they're not going to have learned very much. So you still got to do the work. The brain is shaped by environment and what you do with it. So I think it will be fairly broad, and how long-lasting it will be, it's unclear. I mean, it should be pretty long-lasting. It should change the way the brain works, hopefully. But that's just a prediction at the moment. So we really are kind of ready to go. We're trying to raise the money to do this. This is not the kind of thing you can go to the National Institutes of Health and say, hey, give us $750,000 to build some video games for doing intervention, because they'll say, well, where's the evidence? And we'll say, well, I think it's going to work. And they're going to say, yeah, come back when you've got some real evidence. So we've got to actually develop the preliminary evidence first. But they're protecting the taxpayers' money. That's their job. So we have to look in other places. And it may be that we have found some of that money and we can do it fairly soon. So it's pretty exciting. But at the same token, what I want you to understand is if you understand this notion that the difference has to be bigger before you see the difference, and how the system is working differently, it might give you a way to go away and use this with your kids or the kids who are in your classroom or whatever. Think about how you might change things. If you want to say, is this thing bigger than that thing, our senses probably get two things that are very similar and see if you can tell them apart. Well, that would be completely the wrong way to go. You want to make very big differences. If you want to count things, you want to make them all different colors so they all stand out from one another and have them very spread apart, and start with very small groups so you can understand the process, and then you can get to bigger groups once you actually can comprehend it, and so on and so forth. So hopefully you can kind of draw some of that from this. I have a website here. I didn't put the address up. My lab is called this thing here. It stands for Cognitive Analysis and Brain Imaging Lab. And if you pick up a flyer out there, actually, or you already have one, our web address is on there. But if you write this down, CABIL @mindinstitute.org, that's the web address. And there's a talk somewhat similar to this already up on that website under the Fragile X talks. And in fact, you can look under the 22Q talks. There's actually presentations that I've given, either what we call the family meetings or other places. And I have a huge grant application due in 10 days to keep this work going. So I don't promise to do anything before that, but after that, I'll get these slides ready, and I'll put them up there, and Dan can help me put them up, and they'll be available. So maybe in a few weeks, these slides will be available too if you want to use them. Okay, thanks a lot.
The UC Davis Mind Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.